Great, thank you. Well, thank you so much, Dimitri, and to the Guarantors of Brain for this opportunity. So I'm going to try and give you an overview of clinical and molecular genetics. I'm going to target it largely to adult epilepsy because I understand the majority of the attendees are more towards adult rather than pediatric neurology, but I'll certainly make some comments about the pediatric side where spectacular advances have been made. So I'm going to first talk about the evidence for the genetic basis of epilepsy, which is clinical evidence, the status of gene discovery in epilepsy, and particularly what we like to call the hidden genetics of epilepsy, and then some new, relatively new things, particularly mosaicism, the quest to solve the common epilepsies, particularly GGE, and then polygenic risk scores, which one of the flavours of the moment. I'll briefly talk about precision medicine, but there are a couple of exciting talks that are dealing directly with gene therapy, so I won't touch on that and leave it to Matthew and the others to deal with that. So if I tell you as an adult neurologist that I believe that much of epilepsy is genetic, you'll tell me that's rubbish because most of the patients you see with epilepsy don't have a clear family history, and I'll try and convince you otherwise. So here is the data on basically clinical genetics of epilepsy. These are familial aggregation studies which ask how frequently is there an affected relative in persons with specific disorders, and if the number's 1, it's not greater than controls. And as we're all aware, generalized epilepsy has the highest heritability, and the recurrence risk ratio is about 8. Focal epilepsy, which we used to think of as sort of solely acquired, is much lower, but much greater than 1. It's about 2.5, suggesting that there is in fact a large or considerable heritable component to focal epilepsy. And febrile seizures, the commonest one, but obviously a pediatric syndrome, which I won't talk about much more, is intermediate. This is contrasted or complemented by twin studies, which is the other good way of getting a hold of the genetic component. And here we compare the concordance. That is, if twin 1 has the disorder, does twin 2 have it? And we compare it between the MZ or identical monozygous twins compared to the dizygous or non-identical twins. And for generalized epilepsy, the figures is 0.8, which is incredibly high. There aren't many complex diseases that approach that. And that kind of screams at you that generalized epilepsy is fundamentally a genetic disorder, even though you won't see a family history that often, as we'll discuss. In focal epilepsy, it's 0.4, again, much higher than one might have thought intuitively. And in febrile seizures, it's 0.6, again, in the middle. So those two sets of data match one another. Before the molecular genetic revolution, before the whole genome project, the way we got at this was family studies, and we studied some of these very unusual rare families, and the initial breakthroughs in gene discovery were made, but these don't apply to the majority of patients. And we can think about epilepsy, or any patient with epilepsy, as lying on a spectrum with a contribution of genetic and acquired factors. On the right of the screen are the essentially purely acquired disorders, post-head trauma, post-stroke, post-infection, etc. And epidemiology suggests that about 25% of the patients we see fit into that. On the left are those, what we used to think were the rare single gene epilepsies, particularly manifested by those very unusual large families. And in the middle were the epilepsies with polygenic inheritance, such as generalized epilepsy and some of the familial focal epilepsies. The first breakthrough from sort of the new genetics of epilepsy was that the single gene epilepsies were considerably more common than we previously thought, and I'll explain why that is just now. So with the Mendelian epilepsies, the initial large families opened this door, and the initial discoveries were a group of ion channel subunits, both voltage gated and ligand gated. There are also some non-ion channel genes, in particular LGI1 for lateral temporal lobe epilepsy, and GLUT1 are very important because it's treatable by the ketogenic diet. And these were seen predominantly in large families. But interestingly, we found that what we thought was a pure, nice clinical syndrome, and epileptologists love their electroclinical syndromes, generally all turned out to be heterogeneous. The hidden genetics of epilepsy, the fact that there's much more monogenic or inheritance of all sorts in epilepsy, is due to a number of factors. First of all is the under-ascertainment of family history. Adult neurologists, and I can say this because I am one, don't take good family histories. You take it quickly at the first consultation. If it's negative, you tick that off and you don't go back to it. But the truth is that the adolescent or young adult in front of you doesn't really know whether his uncle or aunt had seizures when they were young, much less his grandparents. And it takes a lot of work to winkle that out, particularly with the stigma that still is associated with epilepsy. Secondly, in disorders with complex inheritance, we expect an inobvious family history. They don't jump out at you as disorders where the thing runs through the family like a golden thread. And this can be for a number of reasons. Sometimes there's a very subtle family history. For example, we did an intense study of new onset temporal lobe epilepsy, looking for patients with intense déjà vu. About 60% of us get déjà vu. It's a normal phenomenon, but sometimes it's just, it's really tricky to decide whether it's physiological or more than that. And we showed by looking at subjects' relatives compared to controls that the really intense déjà vu was much more highly representative, suggested that this was in fact a familial trait. What we didn't expect was the importance of de novo mutagenesis, that is, new mutations. And this is particularly true in the early childhood epilepsies, what we now call the developmental and epileptic encephalopathies. The poster child of this is Dravet syndrome, which is nearly always due to a de novo mutation in SCN1A, the sodium channel gene. But there are now numerous single gene de novo disorders, probably about 200 now, that cause a variety of severe early childhood disorders. And what we used to make a diagnosis in inverted commas of epilepsy and mental retardation, is now a very complex group of largely monogenic, predominantly de novo disorders. And these can be due to copy number variants as well. So this has now transformed the management of early childhood epilepsy. The number of genes involved is enlarging. They involve all areas of the postsynaptic membrane, the synapse itself, and a bunch of other structures. And we're learning a lot about the biology of these epilepsies by these individually rare but collectively not that rare severe disorders, and these are the focus of some of the precision medicine approaches. So that's very important in childhood epilepsy. We think de novo mutations also happen in later onset ones, but these are less well studied. I want to now talk about a couple of new genetic insights, and that's mosaicism. Here's a case of hemimegalencephaly. This is usually or almost always non-familial, a spectacular disorder. And this remained quite a mystery how this occurred. Everybody thought something odd happened in utero, but what is odd that happened in utero wasn't a vascular event or an infection, it was a genetic mutation. And we've learnt that these mutations occurring after fertilization can cause severe and lateralized epilepsy. And hemimegalencephaly turned out to be due to post-zygotic mutations in the PIK3A, AKT3 pathway, which is shown here, feeding into mTOR, a very important gene for cellular growth. And the figure's probably now higher than 30% as we get to lower levels of mosaicism. And this is a gain of function. This disorder is a gain of function disorder in this pathway. So that was also sort of a very important insight that really made us think, is mosaicism more important? Now, turning to a disorder that many of you that work in adult epilepsy surgery clinics will see, here's a case of, here's an example of bottom of the sulcus dysplasia. When you look at this MRI for the first time, you don't see it. When you look at it for the second time, you don't see it. And when you look at it for the third or fourth time, you see this subtle blurring of the grey-white junction here, and it's sort of shown by this cartoon here. And a number of groups now have shown very low-level mosaicism in this disorder. And here's a study from our colleagues at the Children's Hospital in Melbourne, where they were able to specifically look at the bottom of the sulcus and the crown of the gyrus. This patient has a very low-level mosaicism in mTOR itself, in the mTOR gene itself. And these are droplet digital PCR plots showing the frequency of the mTOR mutation. They were able to show that it's it's prevalent at about 5% at the bottom of the sulcus. And you can see these cells at the bottom of the sulcus staining with S6, which shows activation of mTOR, whereas at the crown of the gyrus, no mutant is detected, and you don't see S6 staining. So very small lesions can be due to mosaicism, and these are genetic causes, although they are not inherited, but very important. So that's now a big theme in the understanding of the genesis of focal epileptogenic lesions, particularly in the epilepsy surgery cases. What about that disorder that jumped out from the twin studies, the epilepsies with polygenic inheritance, particularly genetic generalized epilepsy? And this has been a real puzzle to understand this, because it's clearly genetic by the clinical studies, but finding the genes has been tough. I tell you it's genetic, and then you'd tell me, well, most of the patients you see with this disorder, for example, juvenile myoclonic epilepsy, don't have a family history. Correct, they don't. You occasionally will see families like this where there might be one JME subject and they have a sibling with childhood absence epilepsy, or when you dig really hard, there's a grandparent with generalized tonic-clonic seizures, and that might be about 20% of the families. If you work in a country where they have much larger families, it'll be more frequent. And very rarely you'll see a disorder that kind of looks like it might be monogenic, and this would be an example of something like a GLUT1 family. So this is the clinical genetic architecture of generalized epilepsy. And of course, the frequency of the family history depends on the SIB-SHIP size and also on the thoroughness of your history. But there are no known acquired factors for this group of disorders. For counselling purposes, we can say, sorry, have I lost the slides now? Do you see them?
They're still there, yeah. We can see it.
Sorry, I've done something silly to the other thing. We'll manage fine. The risk to family members is about 8%. Okay. Sorry, my slides aren't moving. I'll reshare. Sorry. Okay, we're back in business. So this is a paradigmatic complex disorder, and just to say a couple of words about the way the genetics community is thinking about these complex disorders. There are two major hypotheses for the genetic architecture, and these are reflected by this graph here, which shows the odds ratio of a variant predisposing to the phenotype versus the minor allele frequency, which can get up as just under 0.5. And what we can observe are very rare variants of very high effect with a very low minor allele frequency, and this in its extreme is a classical dominant disorder. Or we see common variants of very small effect, and these are the things we pick up in GWAS. We don't see common variants of large effect that are common. They just don't occur. They're selected for against evolutionary. We don't get them. And rare variants of small effect we just can't detect. So we're chasing these rare variants of big effect or these common variants of small effect. And there are basically two models: the common disease rare variant model, where we've got relatively large effects. There is robust evidence for this acting in generalized epilepsy by copy number variants. They're not that common, but they're common enough and they contribute to the architecture. And we're now looking very hard for point mutations using very large whole exome sequencing projects, Epi4K and Epi25, which I'll give you some of the results for in a moment. The alternate hypothesis, which I must admit I wasn't that much of a fan of for generalized epilepsy, is where it is due to common variants, very small effect size. These are studied by genome-wide association studies, but there's been debate as to, how can these small effect sizes, 1.05, etc., how can that be clinically meaningful? What can you do with this? And people used to think nothing, but now we actually can do something with it with polygenic risk scores, which I'll explain in a moment. So there's an ILAE consortium trying to solve find these common variants, and it took a while to get the numbers together. This represents about 4,000 patients with genetic generalized epilepsy, and a number of genes are showing up on this Manhattan plot. Some familiar ones like SCN1A, some expected ones like GABA receptor variants, and a couple of funny ones. And we are still underpowered with this study compared to many other diseases, but the ILAE consortium is growing, and we expect that a whole bunch more will be discovered shortly. In terms of the rare variants, the first big study was Epi4K, which I won't show you because Epi25, which is larger, has essentially replicated this. And the way this is done is a case control analysis of comparing the frequency of variants in particular genes compared to controls. And where the genes lift off the red line, they're the ones that become interesting, and then they may reach a specific threshold. Now, disappointingly, nothing has yet reached individual statistical significance in relatively large samples, or the samples are growing. But in terms of ion channel classes, in terms of gene channel classes, the ion channels are disproportionately represented. So we believe we're sort of getting close to the truth here, but we still haven't got it, despite some large studies. So we can think about the genes that are influencing genetic generalized epilepsy like this, where I've got a plot of odds ratio showing the genes of very large effect. And here we've got these genes that more or less behave like monogenic disorders, which are very rare and come from those extraordinary families that probably represent less than 1% of GGE. The important one for treatment is GLUT1 or SLC2A1 because they respond to the ketogenic diet. There's also a number of GABA receptor genes that pop up. Here we've got some genes of sort of intermediate effect size and minor allele frequencies of sort of 1 in 1,000, 1 in 5,000, etc. And these are some of the recurrent copy number variations, of which 2 are robustly associated with generalized epilepsy. And down here, with a very low effect size, are the genes that pop up in the genome-wide association studies. Again, as I pointed out, a bunch of ion channels, but some other genes as well. And we expect that as the numbers grow, this graph will fill out. Now, as I said before, some of us used to think that GWAS data was pretty useless clinically. But what is done now is summing these, and this shows some very interesting results, which I'm going to show you in not in epilepsy because we don't have the power yet, but where this is going. So here's a striking single example. It's not high science, but it's cute. This is the polygenic risk score for height, which is, as you might expect, a normal distribution. There have been studies done on 100,000 patients or subjects and looked at their common variants with respect to their height. Here is a very unusual man. He is 7'6" tall, 2.3 meters in the new language. He does not have any disease. He's a very skilled basketball player. Sean Bradley's his name. And where does he sit? Well, he sits out here. sort of 10 standard deviations in the polygenic risk score. Now, this doesn't completely account for his height, but it kind of shows you that this is a good way of sort of ranking this physiological variable. And the polygenic risk scores are made by summing most of the SNPs that you see in these GWAS studies. That's where Sean Bradley is. Now, this is now entering the clinical realm, not yet in epilepsy, but this is the way it's going. So what this graph shows is the risk of a very well-known genetic disease, one that a Nobel Prize was given for. That's familial hypercholesterolemia, the old Brown and Goldstein story. And this shows the probability of coronary disease consequent on your polygenic risk score. So if you don't have a gene for familial hypercholesterolemia, your risk of coronary disease is down here. It goes up with a higher polygenic risk score. But if you've got one of the genes, your risk varies from 20% to 80% depending on the common variants. And the same story is true for cancer genes. Here's breast cancer, showing that the polygenic risk score for breast cancer is influences even if you've got a major gene like BRCA1 or BRCA2. So we're now trying to understand how common and rare variants interact in the epilepsies, and we think we're slowly getting there, but there's still a fair bit more work to do. But this is what's going to happen in the next couple of years. So is this relevant in clinical practice? Well, it certainly is in pediatric epilepsy, particularly in those epileptic encephalopathies. And the closure of the diagnosis for the patient is really important for the families. They stop searching. It allows them to focus on the problem. It brings the families together, and some of these foundations are incredibly powerful for driving research. It stops further testing. They don't need another MRI. They don't need a stereo EEG. They need, as best we can, to tailor the treatment to the disease. And occasionally, or rarely perhaps at the moment, you pick up something that's really curable, like an unrecognised vitamin B6 deficiency. But that, to be fair, is rare. However, it still does change things. If you diagnose Dravet syndrome, you don't give them sodium channel blockers. And that applies to the child with the new diagnosis or the adult that you see with epilepsy and intellectual disability who's been in a home and never had a specific diagnosis and still having lots of seizures. If you see that person and you think they've got Dravet syndrome, test them. And if they do, get them off the phenytoin and the carbamazepine and get them on to the Frisium and the Topamax, and it makes a big difference. Similarly, or inversely actually, with SCN2A and SCN8A encephalopathies, they respond to sodium channel blockers. And that's probably because SCN1A is largely on the inhibitory neurons, whereas SCN2A and 8A are on the excitatory ones. GLUT1 deficiency, ketogenic diet, tuberous sclerosis is increasing evidence for everolimus. It's not a cure for the epilepsy, but it does seem to improve the seizures to some extent. And there's growing interest in the avoidance of side effects. For example, the Stevens-Johnson syndrome with carbamazepine, although that early example is still the most spectacular one of the HLA antigen in Han Chinese, but much more work is going on this, and I think we'll get much better pharmacogenomic profiles in the future. In terms of precision therapies, there's a push now to repurpose licensed drug. That hasn't worked as well as we'd hoped, and there are a bunch of new therapies, which or experimental therapies, which a couple of the further talks will address, and I won't speak to given the time. So to summarise, what have we learned about epilepsy genetics and how do we think about it in the clinic? The role of genetics, in my view, has been underestimated. Mendelian epilepsies are uncommon and familial aggregation is modest, but in many more patients than we think, there are underlying genetic mechanisms. De novo mutagenesis is very important. Mosaicism is important as being very important. Our initial idea that we diagnose very cleverly the epilepsy syndrome and there'd be one gene, one syndrome is completely wrong. Most of the disorders are very heterogeneous, but having said that, the phenotype still gives you direction in terms of where you should be looking. The common epilepsies are probably due to an interaction of common and ultra-rare variation. We're still to completely nail that, but that's where it's looking. It's been a very tough road to walk down, but we think we're getting closer with these very large numbers. Precision diagnosis is now a reality, particularly in paediatrics, but to a lesser extent in adult medicine, that you can get a diagnosis. And in the epileptic encephalopathies, the number's up around 70% now. We're starting to learn more about their roles in the adult epilepsies, but we're not quite there yet. And whilst precision diagnosis is here, precision therapies are still a little way off, but we can see them coming, and hopefully it won't be that far away in your clinic. So our work's been due to a very large number of people, both my group in Australia, but very major collaborations. And I'll leave it at that, and thank you very much for listening.
Thank you, Sam. That's terrific, and thank you for keeping to time. One of the disadvantages of these online conferences, you don't hear the applause from all the audience, but I'm sure they're out there impressed by your ability to cover so much in so little time. Sam, can I just jump in there with a question? So, of course, we're starting to understand the impact of somatic mosaicism for the mTORopathies and a few other genes associated with structural malformations. But what about the ones that are not? What about the ones that might be like ion channels? Do you think that may be contributing to some of that hidden heritability or hidden?
Yeah, no, I certainly do.
How do we go about that? Presumably, we're not going to get the tissue very often.
No, we don't get the tissue very often. We are looking, trying to figure out some clever ways to do it. In fact, paper we got into brain communications, we're looking at CSF for cell-free DNA, which even though lumbar puncture is mildly invasive, and we're starting to get some success about detecting that with that, but that work's still got a way to go. But yeah, look, it's a real problem getting working out mosaicism in the brain without brain tissue.
Right. I'll just read out one question because I think we need to move on. But irrespective of whether your GGE is attributable to a single gene or polygenic risk, do you think there are common pathways that lead to manifestation of disease? If so, at what scale, e.g. neuron level, regional conductivity level?
Yes, I do, but I don't know the answer to the second part of the question. I mean, what strikes me as a clinician is that I can diagnose JME in Melbourne, and you'd make the same call in London, and they'd make the same call in Tokyo or in Nairobi, and it's so characteristic. And yet we're seeing this heterogeneity. It must feed into some common pathway, but we need to figure out what that is. I don't know what it is.
Okay, thank you very much, Sam. We're going to move on. So the next talk